Hi everyone. So today we are going to look at genetic crosses, specifically monohybrid crosses. In grade 12, you will learn about the types of genetic crosses that you may encounter in your examinations and tests. So um, they are the monohybrid and the dihybrid crosses. So in this video, we are going to look at monohybrid crosses only, and we're going to look at how can you solve a problem whereby you have to do a monohybrid cross. So the information I'll be sharing here is based on Mind the Gap study guide for grade 12 learners for life sciences. So let's dive right in. So the first thing to note is that when we talk about a monohybrid cross, we are referring to a type of cross that involves the inheritance of one characteristic. So we are only going to focus on one characteristic. Hence the meaning of the word or the prefix mono. Mono means one. So when we say monohybrid cross, we are going to consider only one characteristic. And when we say dihybrid cross, we are going to consider two characteristics because di means two and then mono means one. So in this video, we are only going to focus on monohybrid cross, which is a type of genetic cross that only involves the inheritance of one characteristic. So the problems that you may have to solve in your tests and examinations are incomplete dominance problems, incomplete dominance, codominance, inheritance of sex, inheritance of blood groups, and sex-linked characteristics. So you need to be familiar with these types of crosses, like the crosses that involve these kind of situations, in order for you to be able to answer all the different types of questions that may be presented to you, because they fall under these categories that are mentioned here. They fall under these categories. So you can also watch a video whereby I explain the degrees of dominance, which are complete, incomplete, and codominance, on the link that I have provided here. I have used an authentic example to explain the different degrees of dominance that we have, so that you can be able to understand what they mean by complete, incomplete, and codominance. So you can just uh, put this link on your browser, and then it will take you directly to the video. And when you watch the video, please don't forget to like and subscribe and comment and even share, to help me grow, so that I can keep posting. I can stay motivated to keep posting more videos, helping you unpack these different things that are kind of difficult for us. So for you to solve genetic crosses, like in grade 12, specifically monohybrid crosses, you follow this format. This is the template that you are going to use in order for you to solve your genetic crosses for monohybrid crosses. Like, you're going to use this format, or you can use the Punnett square format. So for just putting this format as it is, you will just get 2 free marks. This is what the guy here says, that when you just put this format, you are going to get free 2 marks. So even if you don't know how to solve the problem, at least by putting the format or just the template, then you will be able to get free 2 marks. So make sure that always you put the format or the template before you start putting everything else that you are given. So you need to make sure that you include those. And then with the Punnett square one, you you mention the phenotype, then you mention the genotype, then after mentioning the genotype, you write meiosis, then you draw the Punnett square, and your Punnett square must have 3 columns and 3 rows. So you draw the Punnett square that has 3 rows and 3 columns, and then you write gametes here. Then you will, on this side, you will show the gametes of the parent on the left hand side, and then on this side you will show the gametes of the parents on the right hand side, and then you start crossing them. matching them. Like if let's say here it's capital B and here it's small b, then you're going to put capital B first, and then you put small b second. Like always when the characteristic is the same, you put the uppercase one first, then followed by the lowercase one. Doesn't matter which one comes first, the uppercase one must always be the first, followed by the lowercase one. So you do that, and you match them uh, based on what is on each block. Yeah, you match them based on what is in each block. Fine. So let's just look at an example. So in this example, they are saying that in rabbits, the dominant allele, capital B, produces black fur, and the recessive allele produces white fur. Use the genetic cross to show the possible genotypes and phenotypes of the F1 generation for the fur color. For the fur color, if two heterozygous parents are if two heterozygous parents are crossed. Yeah, this was not typed well, but yeah, the question just says use the genetic cross to show the possible phenotypes and genotypes of the F1 generation if two heterozygous rabbits are crossed. So you are going to look at the parents that are both heterozygous for the characteristic of the fur color. So step number one, you need to look at which trait or characteristic are you considering here. So in this problem, we are looking at the fur color. That is our characteristic or our trait. Then for the letters, you are given capital uppercase B and lowercase b. So those are the letters that you're going to use, and usually in your in grade 12 they normally give you like the letters that you are going to use. If it happens that you choose your own letters, make sure that you choose the letter that its uppercase and lowercase differ. Like you can be able to easily tell the difference. For an example, with C, you can't like the uppercase and the lowercase look the same unless if you just make the other one bigger than the other one smaller. So it is advisable for you to use letters that the uppercase and the lowercase differ, so that you can be able to tell the difference. And then if you're looking at one characteristic or the same characteristic, you need to use like the same letter for that particular characteristic. So if you're looking at the fur color and we're saying that we are using B, for the dominant and for the recessive, you use B. Like for the same characteristic, you need to use the same letter. If we are using B, then you have to make sure that you use B for both recessive and dominant, depending on which one is the dominant and which one is the recessive. Then the dominant one is going to take the capital letter, then the recessive one is going to take the lowercase letter. Then as you can see here, they have already told us that black is dominant, so that's why you have the uppercase B for black, and it is dominant. Then for the recessive one, we have white, and which we are assigning lowercase b because it is recessive. Then when we look at the genotypes of the parents, we have oh my goodness. Okay, both parents, according to the problem, are heterozygous for the characteristic. This was a mistake. It was supposed to also be heterozygous. So it was supposed to be heterozygous black as well here. So that's a mistake. Then you put your template, and then after putting your template, then you write the phenotypes of the parents, then next you write the genotypes of the parents, then you write meiosis, then gametes, then you show us the gametes, and you have to separate these things. Like you have to separate the gametes using lines. Let me show you how. So you're going to separate the gametes just like this. So yeah, that's how you separate your gametes. Then you come to the step of fertilization. Then obviously after fertilization, you will have the genotypes of those offspring. Then you are going to take the first gamete from the first parent, then you put it here. Then you take the first gamete from the second parent, then you put it here. Then you take the first gamete from the first parents, then you put it here. Then you take the second gamete from the second parents, then you put it here. You take the second gamete from the first parents, you put it here. Then you take the first gamete from the second parent, then you put it here, and you do the same for the second gamete for the first parents and the second gamete for the second parent. And then if you can check here, whereby we first had the lowercase b in the first parent, and then on the second parent you had an uppercase. You can see that when we wrote the genotype, we first began with the uppercase, then followed by the lowercase. So you need to be careful of that and make sure that even if the lowercase comes first, you first put the uppercase, then followed by the lowercase. Then this one is gonna just, it's gonna be like this, and you have to include these lines. They didn't include it in Mind the Gap, but according to curriculum advisors now, you have to like include these lines. They will like show you a little bit of logic. Then looking at our genotypes, you can see that we have three parents with black fair color, and I mean, no, not parents, we have three offspring with black fair color and one offspring with white fair color. So that is just what is written here. So you can just decide that you put it this way, maybe three times three times black and one times white. So sometimes they may ask you to give them the phenotypic ratio of the black, maybe the black uh, offspring. So obviously it's gonna be 3 out of the 4 multiplied by 100, no, not the phenotypic ratio, that like the percentages of the percentage chance of the offspring being black. So it's gonna be 3 out of 4 multiplied by 100, and the phenotypic ratio here is 3:1 because you have 3 black and 1 white. So it is 3:1. So you just put it like this. Oh. It is 3:1. Like you just put the ratio. This pen is disappointing me, but you know how to put the ratio, and if you don't know, you can always feel free to comment and ask me to show you how you can put the ratio. Then when you are using your Punnett square, as I said, you put the phenotype, the P1 phenotype. Remember, the P1 and the F1 are always important. You need to ensure that you put them as well to get those 2 free marks. So you put P1, and then you put the phenotype, the genotype, then the meiosis. Obviously, meiosis will result in the formation of gametes. What are those gametes? Those gametes are here. Here they are shown, the gametes. Here you have your gametes, and then you match them, then you match them all the way until you have the last genotype of the offspring. Then you write the phenotype because you've already mentioned the genotype here, and yeah. Here you write fertilization, so you must show us that fertilization is occurred here, therefore you have the genotypes of the offspring. So you write fertilization here. Uh, Yeah, I'll just write that, you will know what it means. So you just write fertilization, then you will have the genotypes of those offsprings that you have been given, or you have worked out. So we will we will have three black and one white. So you can see that these things produces the exact same things. It's just that you will use the one you prefer, either the Punnett square or the other one that we did previously. So this is how you can deal with your monohybrid crosses. So if you still need clarity, you have some questions and you want me to help you with, just type in the comment section what you need help with so that I can attend to it as soon as possible, as soon as I can, so that you can be able to understand these things. I hope I made sense. So thank you. Don't forget to like, subscribe, and comment.